Okay, so this is my biweekly update for Friday the 27th of January. So, regarding progress from the last biweekly update, most recently I shadowed Nathaniel on two separate occasions, and I watched him perform a media change this past Tuesday, and then a DNA precipitation immediately afterwards. Today, I watched him perform a viability count immediately after a lipofectamine transfection. And this was for the OATP project he's working on, and I will most likely continue shadowing him into the future. And in the past week and this week, I began conceptualizing what I'm going to do for this docking workflow, specifically how I'm going to change the existing docking protocol to accommodate um, these these antibodies, these SC fabs, and how I'm going to increase precision because these docking results will either inform wet lab conclusions or they'll be used in tandem with wet lab results. So precision here matters more, so I'm trying to do everything I can to increase the accuracy of the docking. I met with Annie today to discuss the assembly of the sequences and what I'm going to do for the docking. We found all the components of the sequences with the exception of the constant heavy, which I believe you sent in Teams immediately after we met. So we now have every component that we need. So the constant heavy right here. And then all of the other ones we have. And the assembly will be performed in the following order. So variable light, constant light, a space for a linker, variable heavy, and then the CH1. The linker, we're just going to put the linker from that was used in previous AlphaFold runs because we're considering that the linker is not going to really affect the antibody paratope in the docking. So it's just there so AlphaFold and Rho beta and iTaser will be able to express it as an SC fab because we have doubts about its ability to express it as a fab. It needs to have a fully connected sequence to our knowledge, but we're going to try it without a fully connected sequence too, just to make sure that it can possibly do that. And yeah, I was wrapping up those applications for the U of M and Van Andel program. Okay, so the ultimate docking plan that I've conceptualized. Okay, first we're going to have the sequence assembly, which I showed in the spreadsheet, and the homology model construction with AlphaFold, Rho beta, and iTaser. If we get to iTaser, but yeah, the plan is to have a comparison between AlphaFold and Rho beta. After the model is created, Rosetta Relax, which will energetically minimize the homology modeled structure in order to help it accommodate to Rosetta's force field. All Rosetta protocols recommend you run this in order to make the structure better suited to Rosetta. They recommend you run this on structures not obtained from Rosetta, so AlphaFold fitting that criteria. Then we're going to do conformer generation. So traditionally I've done 100 conformers for each ligand in the COVID project, but because GD2 is much larger than any of the ligands in the COVID project, it's more complex, and I want to be far more precise in the docking, I'm going to use more conformers than 100. I'm thinking about 200, but I'll need some literature benchmark that shows that one, docking accuracy increases with increasing number of conformers, and two, I don't know, based on number of rotatable bonds in a molecule, what is the optimum number of conformers, say, that you should generate. Maybe there is such a formula. I vaguely recall seeing something like that in a paper we used for the COVID project, so I will refer back to that. But the general trend was that docking accuracy increases with increasing number of conformers. So first and foremost, I'm going to need the actual NHAC GD2 structure, or it might be, 9 NHAC GD2 and a 9 OAC GD2. I'm probably going to end up docking both of them. So the one I made in iBabble was pretty significantly different from this one that I saw in Shufei's paper that we referenced in the methods chapter. And so they actually have a digital structure of 9 NHAC GD2 and 9 OAC GD2. And we're just going to use this because they had actual results with this structure. They published this structure. This is the official SDF digital structure of 9 NHAC GD2. So this is what we're going to use. I emailed Po-Han Lin from Shufei's lab today after speaking to Zara, asking if we can have this SDF structure to use in our docking, again, for precision. Then, obviously, Rosetta protein ligand docking. I don't know if these GD2 structures fit into the category of glycans, and if glycan dock would be good for them. If I can figure out glycan dock, I'll probably implement it. Glycan dock doesn't work with standard conformer generation. It uses the GA ligand dock algorithm, in which it spontaneously generates conformers in the protein ligand interface rather than using input conformers like in files like BCL. You generate 100 conformers, you input all 100 into the docking workflow. With GA ligand dock, the conformers are generated there. It's supposed to be better, but okay, for the actual docking, I'll select as many coordinates as I can within the antibody paratope regions and the CDR regions to make sure that I sample the entire CDR region with the docking grid size. In future updates and in the paper, there's going to be a paper showing the exact regions in the antibody paratope, a very, very detailed figure showing spherical regions that were sampled in the antibody paratope, and it's going to have the entire CDR region sampled, showing that we truly analyzed it thoroughly during the docking. And instead of generating 1,000 outputs for Rosetta docking, I'll generate more than that because, again, I want to be accurate. This project sort of demands increased accuracy compared to the COVID one. So probably 3,000, 5,000. And timing shouldn't be an issue here because there's only one ligand, well, two, 9-OACGD2 and 9-NHACGD2, and there's only 10 structures, so this shouldn't take a lot of time on HPCC. Ultimately, the docking results, it's a good idea to perform a comparison between the rho beta and the AlphaFold results. If we get ITASR results too, So compare the RMSD between the different homology models and then show the differences in the best binding occurrences for each of the homology model categories. So the best binder for rho beta, best binder for AlphaFold, and best binding for ITASR. For the overall lowest energy pose, it might be interesting to do an MD on it. As you mentioned, Michael Feig uses MD on HPCC, and we might be able to access his workflow there. And just get a general ligand RMSD versus time graph, which is good for determining stability in a specific binding pocket over time. And then usually with MD, what I've seen is they discuss specific interactions like we did in the COVID paper at the end, hydrogen bonds, pi-pi stacking, salt bridges, etc. And it's usually used to reinforce the findings of the MD that the ligand is stable in its site. So the RMSD is constant over a period of time. That means something is holding the molecule there. What interactions are there, you can then name them. And then, writing the paper, integrating this with the laboratory findings. Questions. Well, it seems that we're putting humanization, affinity maturation, design sort of on the back burner for now. But like, how will these eventually be integrated into the workflow and the paper, if at all? And how will the paper that all these results go into, the ones that Annie's working on as well, be structured in terms of content? And this is a paper I read recently.